I'd like to welcome you and thank you for joining us this afternoon for the joint webinar between Symbiotics and PAML, Cancer Genomics, Clinical Applications for Next Generation Sequencing. My name is Eva, and I'm your host this afternoon. Housekeeping items before we get started. When you did join, a pop-up box should have opened with the handouts. You're able to click on those and download them to your computer if you'd like to save copies. One out of today's presentation, and the other one is the evaluation that we'll be asking you to send at the end of the presentation. Difficulties, please KCHANDLER@pam.com. Chandler, and she will be able to help you. This afternoon, on the side of your screen, there should be a panel. At the top, there's a blue bar that says Q&A. If you left-click in the white box at the bottom of that panel, you can type in a question and send it in. You can do that at any time during the presentation. We will be saving those and keeping those till the end, and Dr. Harry will answer questions at the end of the presentation. Also, to receive PACE credit, you're completing the evaluation, and we'll keep the webinar open for about 10 minutes after the presentation so that you can download any documents that you need. The site said we will just need to download it and then submit it, and you will receive a PACE certificate within a couple days. Also, if you do just track your own CE, you can email Kathy Chandler, and she will send you a certificate of completion. That's just something if you keep that for your own records. And our employees that are joining today will be able to receive our continuing education through CE Manager. Now, welcome our speaker for this afternoon. Of molecular pathology at Symbiotics. Prior to joining Symbiotics, she was the associate professor at the University of California, San Francisco. Dr. is board certified in molecular genetic pathology and pathology. She received a fellowship in molecular genetic pathology from the College of Virginia and her fellowship in cytopathology from the National Cancer Institute in Bethesda, Maryland. Dr. Harry, thank you so much for joining us this afternoon and sharing your expertise with us.
To review the learning objectives, at the end of this presentation, participants should be able to understand the role of next-generation sequencing in cancer. You will know which genes we have validated for clinical use. Should be able to interpret the NGS test results and integrate these results with other tests to determine the best treatment path. Any commercial financial relationships with any of the interests discussed in this section. First of all, I'd like to give you a little background about Symbiotics. Symbiotics is a national AP reference lab. The lab was formed in 2013 and launched in February of 2014. It's a 50-50 joint venture between PAML and Cellnetics. It's pathologist-led and has a collaborative focus. Services we offer include flow cytometry, immunohistochemistry, fluorescence in situ hybridization, cytogenetics, and anatomic pathology consults, in addition to the molecular oncology program, which I'm currently leading. The specific topic we're talking about today has to do with next-generation sequencing, and this is the most recent thing that we have developed and started offering here at Symbiotics. So the first question that comes up is, when we speak about tumor genomics, what do we really mean by genomics? A term that you hear tossed around a lot in medical communities and even in the popular press. This is one of those word diagrams for what people think of when the word is mentioned. So you can see that there are lots of different concepts out there for what we mean by this. Guidance from our friends on the internet. The actual definition of genomics is the branch of molecular biology concerned with the structure, function, evolution, and mapping of genomes. And if we look at it this way, we've been talking about genomics for many years, right? It would give you the impression that this is nothing new. However, if you look at this graph at the bottom where we look at the use over time for the word genomics, there's a dramatic increase right around the year 2001, and it goes up logarithmically all the way to where we are now. This is that in 2001, in February 2001, the first sequence of a human genome was published simultaneously in Nature and Science, and at the time this was truly a landmark achievement. Our first human genome was sequenced. This process took about 5 years and cost $70 million. It was accomplished by using several different centers simultaneously that were literally rooms just packed full of capillary electrophoresis instruments. So if you can see this slide now, this is showing just one of many centers that were out there, and these instruments were high-throughput capillary electrophoresis sequencers that were going round the clock for 5 or 6 years. If we were to do the same thing today, it would take a similar amount of time to sequence one genome using one instrument and would cost us about $30 million. Sequence of the human genome, lots of technologies emerged on the market to try and capitalize on this information that we now had. The most well-known of these is probably the Affymetrix array. The way this technology works is that there are lots of oligonucleotide probes fixed onto a chip, and then we have labeled DNA or RNA fragments that hybridize to these oligo fragments, and you can see, if it's hybridizing more or less, and you can actually measure these fluorescent signals. This technology is relatively expensive. It's time-consuming to operate, and at the end of the day, you really get one genome. You get one SNP array or, one more specific array. You're really just looking at one copy of what it is you're looking at. So people were actually running these in duplicate or triplicate and using it mainly for research. It is in use clinically, most notably in the Veracyte tests that we do for thyroid cancer. So there have been some clinical applications, but it really was not an ideal technology. Technology that came out is what we're going to talk about today, and this is called next-generation sequencing. So we know, intuitively that this is really important. We hear it talked about a lot, but what are the specific differences? Well, compared to traditional Sanger sequencing using that traditional capillary electrophoresis machine, next-generation sequencing is fast, it's more accurate, it's relatively cheap, and it's scalable. So by scalable, what I mean is we can look at just targeted sequences like we look at in our assay currently. We can look at the entire exome, in other words, the entire coding region of the genome, or we can look at the entire genome. So we target it to the use that we need it most for. Also, this technology is better able to detect heterogeneity. That means if we have just a minor portion of the DNA that has a mutation, we're able to pick that up with this method. If we look at traditional Sanger sequencing, we need to have at least 50% of an abnormal allele to pick it up. But with this technique, we can have a very small percentage and be able to detect it. When we're looking at tumor samples, this is particularly important because usually our sample isn't going to be 100% pure, for one thing. We may have 50% tumor content. And the thing is, we know that solid tumors are heterogeneous. In other words, there may be a minor clone that has a very important mutation, such as a KRAS mutation or an EGFR mutation. So we need to be able to detect this. The top three are relevant, that it's fast, accurate, and relatively cheap, is this principle called massively parallel sequencing. So what we mean by this is that instead of sequencing one strand, like we were able to do with traditional Sanger sequencing, we can sequence 30 million strands simultaneously and then rearrange them to create our final sequence. So if you think about this intuitively, it not only means that we can look at large numbers of targets, including entire genomes, but we can look at each target many, many times in one assay. cells. This one at the top is a HiSeq flow cell signed by James Watson, interestingly. This is kind of the top-of-the-line, biggest application instrument made by Illumina. This cell down here goes on a MiSeq. This is what we're using for our assay, and you can see that it's much smaller. I should say this is the one that fits 30 million fragments, so you can imagine that this is dramatically increased. The advantages to using a MiSeq are that it is faster and it's more accurate currently than using the HiSeq, and I'll go over that in a little bit more detail momentarily. The concept of next-generation sequencing is similar to that used in the Human Genome Project, something called shotgun sequencing. And what that means is we're going to break up the DNA into smaller pieces, we're going to sequence those smaller pieces, and then we're going to align them together to make a larger sequence. As I mentioned a bit ago, in this technique, we're able to sequence millions of small fragments simultaneously rather than sequencing each one of these individual and then painstakingly fitting them back together. On a basic level, first we fragment the DNA, so we break it into fairly uniform pieces. Each one of these pieces is called a read length. On our assay, they're about 200 base pairs long, and the size of these varies depending on the assay and the technology. We label the ends. The blue piece at this end and the purple piece at this end are called adapters, and they each serve a slightly different purpose. The blue adapter down here on the bottom enables our fragment to affix to the flow cell, and the purple fragment is both a PCR primer and a barcode. So one thing that we can do is label each sample individually with one of these purple primers, and it has an embedded DNA sequence that serves as a barcode. So we can actually mix several different samples together. In our particular custom designed assay, we can mix up to 16 samples together in one run on one flow cell, and then still be able to separate them out very accurately based on this barcoded sequence. So we fragment the DNA, we label the ends, and then we fix it to the flow cell. So this is down here to these little spots. Cluster generation, and this is basically PCR, where this fragment kind of bends over, and the primer attaches to this, and then it multiplies, and then it separates, and it bends over again until we have these little clusters of identical read length fragments. This is important because then these signals will be amplified when we go through all of our cycles of sequencing, so that we'll have little clusters that are representative of the same fragment. By the time this process is done, all 30 of those spots on the flow cell are filled up, and we're ready to sequence, and each fragment should look something like this. So this is a thing that's truly unique about next-generation sequencing and has really made this technology possible, something that has made it so revolutionary. This is where we can actually sequence each one of those fragments individually so that we have the entire sequence for each of them. The way this is achieved is a technology that's kind of a spin-off from that traditional Sanger sequencing. So what happens is we have a reversible dye terminator base that we add to the next strand of our sequence, similar to what we would do in Sanger sequencing. So this dye gets added and it stops because it's a terminator dye. Our camera goes in and takes a picture and notes that there's a T here, and then we can actually cleave the terminator portion off of that base and make it so that our strand here can accept another base. So the next one comes along and it's a G. We take a picture, we cleave the terminator sequence off, and we move on to the next one until we've sequenced every in the strand. Out there. The one that I just showed you is the Illumina technology that we're using. The biggest competitor is something called the Ion Torrent that comes from Life Technologies. And the method of detection is a little bit different, but it's a similar technology where we have a terminator base that has the terminator cleaved so that you can go on to the next base. This technology, the strand is fixed to a chip that can detect changes in pH, and as each base is added, it can detect a change in pH. So in this system, you would wash the entire array with an A, and the ones that actually added in will have a change in pH. Then you wash the T, then you wash the C, then you wash the G. So every time there's a change in pH, you know something has been added. If you have multiple bases in a row, if you have 2 or 3 A's in a row, you get a bigger change in pH because you have more hydrogen ions released. This technology is slightly less accurate than the Illumina is because of this change in pH in regions that are repeated. So it's less accurate in accurately determining ranges of repeat bases. It also is a little bit less accurate in determining insertions and deletions where you have little pieces of DNA that are either added or missing. However, I will say that both of these companies have been rapidly increasing the quality of their technology, and both are really fairly similar currently. Sequences figured out, we need to reassemble them. So basically all of this needs to turn into all of this. There are actually 2 different ways to accomplish that. One of them is the de novo method, where we would just take them all and figure out which pieces match and kind of line them up on top of each other in this fashion so that we could then produce the sequence down here. The other way is actually called resequencing, and what we do in resequencing is we have the known human genome sequence that we're comparing to, and we line these up in relation to that sequence and then determine which areas are different. Resequencing is what we use and what I think almost everybody uses, unless you're looking for some new organism, you know, a plant or a bacteria or something where you don't know the entire genome. The de novo method requires a lot more IT, requires a lot more bioinformatics analysis, and it's prone to errors in those same regions of repeat or insertions or deletions. We can do targeted sequencing. In other words, we can only look at the regions that we're specifically interested in. This is actually what we're doing in our assay at Symbiotics. Selection can be accomplished actually by 2 different related methods. What we do is do PCR of the regions that we're most interested in. However, you don't necessarily need to. You can just remove the amplified fragments using magnetic beads. We use a combination of both PCR and magnetic beads to remove the fragments that we're most interested in. Targeted sequencing, in other words, only looking at the area you're most interested in, allows for more depth of coverage. So in other words, if you're only looking at the regions you're most interested in, those are the regions that are filling up all those spots you have on the flow cell, rather than filling them up with a lot of other stuff that you may not be interested in. Targeted sequencing definitely simplifies the data analysis, because what happens in next-gen sequencing is all data that you get at, you need to look at, because there may be something important in there. And while a lot of really great tools, we have a lot of really great programs and analytic tools, it still does require a lot of human input and a lot of interaction to sift through this data and analyze it. I want to emphasize that targeted sequencing is still a huge amount of information. You know, if you think about it, it used to be we would look at 2 or 3 exons with Sanger sequencing. You know, now we're looking at 68 genes on our assay, which adds up to a total of 700 amplicons. And we're looking at them in depth. So we can see minor clones, we can see heterogeneity, we can see lots of different findings. So the amount of data that we're getting is still massive, even with targeted sequencing. A few things for a laboratory to consider when choosing a platform. First would be the quality or accuracy of reads, and this relates back to something called a Fred score. The Fred score is a mathematically determined value that was based on Sanger sequencing and has been kind of translated to next-generation sequencing. This is a little table for how to interpret a Fred quality score and translate that to a numerical percentage that makes a little bit more sense. It's a logarithmic scale, so moving from 10 to 20 means that you move from having a probability of 1 in 10 incorrect base calls to having 1 in 100 incorrect base calls. And then you can see what the percentage that the base you're looking at is accurate correlates to based on this probability and this Fred score. For assay, every has at least a Fred score of 50. Data that we actually analyze, and most of it actually has a Fred score of 100. So here it says 100%, but of course, you know, you can kind of guess from this table that it's actually 99.999, and if you go up to 100, it would be 99.9999. So think about it. This sounds ridiculously high for accuracy, and you think there probably isn't much difference between a Fred score of 30 and 100. And while this is not an illogical way to look at it, it also diminishes the importance of these Fred scores in next generation sequencing, because we're looking at so many different data points, because we are looking at 68 genes, significantly higher number of exons, and a much significantly higher number of amplicons. The more points you look at, the higher your accuracy rate needs to be. Illumina MiSeq achieving a Fred score of 50 or 100 is not difficult, and most of the currently available platforms do have similarly high Fred scores. The second thing to consider is depth of sequencing. I kind of alluded to this a little bit earlier, but let me show you in detail what I mean by that. Top of the slide we're looking at, this is one little segment of sequencing data, and if we look at this, this is our reference normal DNA, and this is the tumor DNA. So in this picture, we have 4 sequences below normal, and we can see that right here there's an abnormality. There's T instead of a G. So we wonder, you know, is that real? Is that like a true somatic mutation? This could even be a germline mutation, right? This could be a patient who's heterozygous at this spot, but it's really hard for us to tell if we only have 4 sequences. And you also can see this over here, this little G. You can wonder if that's also real. It could be a somatic mutation, right? In this sampling here, it's 25%. There, where we have slightly more depth of sequencing, we can see that the T here is definitely real. This winds up being about 80% of our DNA, and this could be your tumor content. If you have 80% tumor content, this is 100% of your tumor DNA. So down here where we have more depth, we can tell that this T is definitely real. We're also picking up a G over here and a T over here, and it turns out that that one single G over here is probably kind of a fluke, because we just saw it once. Say we have at least 250 for depth of sequencing and go all the way up into, you know, 6,000 or 7,000. Our average depth of coverage is 2,000 over the entire range of our assay. Consider as coverage, and that's part of what I just alluded to. You want to look at everything that you intend to cover and make sure that you have adequate depth of sequencing in every single area, because if you're testing for things, particularly if we're using it clinically, you want to make sure that you can return a valid result for everything that you're looking at. So you want to make sure you have adequate depth of sequencing over the entire range of what you're testing for. This is actually another strength of the MiSeq panel, is that there is more uniform depth of coverage across what you're testing, but again, you know, most of these methodologies are getting to be fairly similar. The issue is throughput, and here's where that ion torrent instrument that I showed you earlier definitely has an advantage. That method is faster because it doesn't have to take the picture between the addition of every base. So that definitely is the fastest technology currently. You know, speed does come at somewhat of a price, and the actual amount of DNA that you can sequence on one run is less on an ion torrent instrument. So these are all things that you need to balance off depending on what your intended use is and how much you want to spend per assay. You also need to take into consideration data analysis. This is a huge component, and the various stages of data analysis are handled by different software programs, and the heating companies all offer their own programs in addition to programs that you can get externally. People often ask about the actual numerical comparisons between platforms, and currently this is what I found looking at the most recent information. However, I have this little guy down here to indicate that these types of comparisons are definitely moving target. These companies are actively in competition with each other, and they're really striving to improve this technology. So all of these numbers get better day to day. The things that people typically think about are the sequence yield per run. In other words, how much sequence can you do at one time? The MiSeq can do up to 15 gigabases, whereas the Ion Torrent, the 318 chip that most people are using clinically, can only do up to 2 gigabases on one run. PacBio is another system where you can actually do a single-strand long synthesis. So this is actually like a third-generation sequencer that I think is very interesting, but the technology really isn't quite there, as you'll see by some of the other numbers here. It can sequence up to 100 megabases on one run, which doesn't sound like a lot, but again, this is a single strand. So this is one continuous strand all the way through 100 megabases. So that part is actually more impressive, but not entirely useful clinically right now. Length on Illumina MiSeq, you can go up to 300 bases, and on Ion Torrent, you can go up to 400. On PacBio, average is 1,500 bases. These are pretty competitive, and again, these two companies are really striving to increase these. These numbers are not entirely relevant depending on how you use the software to reassemble the sequences. Error, again, is slightly higher with Ion Torrent compared to MiSeq. The runtime is much longer on MiSeq than it is on the Ion Torrent. Both systems can do paired reads, and what this means is they can read the DNA sequence from each end so that you can be sure that you sequence the region in the middle that could be missed by going either direction. Cannot do paired ends. DNA requirements is what people ask about most commonly, and while Ion Torrent has really staked a lot of their reputation on being able to work with extremely small amounts, I would argue that both of these systems are able to work on extremely small amounts, and that the difference between 10 nanograms of DNA and 50 nanograms of DNA is not huge. The bigger determinant of being able to use extremely small quantities of DNA is fixation and the actual quality of the DNA. At these very low levels, if the quality is very poor, it's going to be difficult to get clear results. The PAO instrument, of course, needs a much larger amount of DNA. Cost is a little bit more for the MiSeq than for the Ion Torrent, but the sequencing cost per gigabase sequenced is less on the Illumina versus the Ion Torrent. The PacBio, of course, is much more expensive in both areas currently. Our useful concepts include coverage, is the entire area of interest sequenced, depth, how many times is each base sequenced, reads, so what is the score for the variant that you're interested in. You want to make sure that it's, at least 50 and hopefully 100. The variant allele percentage, how much the DNA has the variant. This is important because, we want to see if it involves all of the tumor content that we have or if it's only a portion. And we want to make sure that it's enough that it shows up above background noise. So we do see some variants that show up at 2 or 3% routinely and uniformly. When the DNA quality is very poor, we see 2 or 3% variants across everything we're sequencing. So pay particularly close attention to those variants that are occurring at these very, very low percentage levels. And we definitely do that in our assay here. Do not filter these out. We look at everything down to 2 or 3%. And if it looks like something that's a known pathogenic variant that we think may be important clinically, we'll frequently go back and try to detect it using another method, using PCR or Sanger sequencing or some other method before we can convince ourselves that it's just background noise. So this is a useful concept to recognize. So the variant is real, we need to determine if it's significant, and some of the different computer programs and concepts that we use to determine if it's significant are listed here. The first one being the effect of the mutation on the protein. So if you remember back to your molecular biology training, each DNA mutation is going to correspond to an amino acid change, and the amino acid change in turn will correspond to an effect on the protein. So you can have a missense change, you can have truncation, you can have elongation. Those are the main ones. You can have insertion or deletion. first thing we do is see, what effect is it going to have on the protein. Then we need to decide what protein do to the tumor. So, this requires knowledge of what the protein does in the body, what the protein does in a tumor, and what's been documented in the literature. Because we can speculate that if we have truncation mutation in a tumor suppressor gene, that's going to be causative of a tumor. It's going to help the tumor grow. But until someone has documented that, either, at least in a study and hopefully in an in vivo study, we don't really know that for sure. So these are some of the things that we detail in our report. We detail what is the effect of the mutation on the protein and what is the functional effect of the protein in the tumor, and we relate that back to the literature. So now we need to look at is what is the incidence of this variant in the population, because some of the things that we detect will be present in the germline DNA, and they could just be common polymorphisms. There are so many differences in our genomes that are just variation from person to person, and they don't really cause any particular problem, and they will not be important to the tumor we're looking at. Some of the databases we look at to determine that include ClinVar, DBSNP, and a couple of other databases that look at common polymorphisms. Some of the databases that we use to look at whether or not the variant is important for a tumor include COSMIC. COSMIC is where people report variants that are important in tumors. COSMIC, I have to say at this time, sounds more helpful than it is because this database is still being built. So often we'll find our variant in breast cancer, but when you actually click through and look at the details of where it was found in maybe one or two tumors and not really in a public study. So we do look pretty deeply into the meaning of all these variants before we decide to exclude them for sure. And I will say that at this time we're including everything that we think may be relevant. At this time, we have a polling question. Karen, if you can put that up. About 20 or 30 seconds to answer the question. Question is, depth of sequencing refers to ABCD. Hopefully you can see the answers. So it's ticking. You're looking at the question, I want to mention that at any time you can put in questions under the Q&A section over on the right side of your screen, and I will have a few minutes at the end to answer all of those questions.
Questions are showing, but just at this time, just go ahead and answer the first one, and I'll put the second one up later on in the presentation. And then just go ahead and click submit at the end to submit your answers. So, okay, when you're ready to move on, I'll go ahead and show the results.
The question was, depth of sequencing refers to the number of times each variant is sequenced. So this is the diagram that I showed a little bit earlier. And this is a concept, again, in looking for somatic tumor mutations, particularly. From there. Moving on, looking at solid tumor genomics, which is what we're talking about here, we all probably know by now that there are clinically relevant mutations used both for prognosis and for treatment. There's a great deal of interplay between mediators, and of course this is a rapidly expanding field. Lots of research studies are looking at whole genome or whole exome sequencing and doing correlations between these findings, and almost every company is looking at these techniques to do drug development. A common example that probably most of us are aware of is this drug vemurafenib that targets the BRAF V600E mutation. And this is the classic picture that was shown in a New York Times article that I think really kind of captured everybody's attention to this area. What we see here is a patient over here on the left that melanoma literally all over the body. Every black spot here is melanoma on a PET scan. You can see it even a little bit up into the brain, definitely the skull, lymph nodes all over the place. Adding the vemurafenib, the the V600E targeted drug, the melanoma is almost entirely gone from the body. So, this is really just a striking, dramatic response. The downside to this story, as probably some of you know, is that this effect doesn't last, unfortunately, with BRAF. The tumors do come back. Is because we know this is a targeted therapy looking at BRAF, and here we're looking at the diagram of all of the mediators that we know are involved in melanoma. Because we know that our drug is targeted at BRAF and that somehow the tumor is getting around the inhibition of BRAF, they were able to move on to the next trial and pair the anti-BRAF drug with an anti-MEK drug. Combination of a BRAF and a MEK inhibitor together are proving to be really effective with melanoma and are prolonging that response. So the ultimate goal for us in doing this type of testing is to be able to target as many of these pathways as possible. There are a lot of mediators in our assay that actually target this pathway over here, including PI3K kinase, AKT, and mTOR. There are many drugs in this pathway, and we have all of these mediators in our panel. So, ultimately, P53, down here, we could target many of these mediators in order to completely stop the tumor. Pathology is involved in personalized medicine many points along the way. We're right here at our NGS assay, and this is in the area of diagnosis, prognosis, and response to treatment. We have these other techniques at our disposal. I mentioned some of these earlier, just basic morphology, flow cytometry, immunohistochemistry, cytogenetics, FISH, PCR, and CGH arrays, which is arguably a better technique for looking at copy number variation. However, we also can get involved earlier on looking at risk factors, and we will be developing a cancer risk assay using the same technology to be available next fall. So, in many ways, it's good to identify your risk early and do prevention. But really, the most important place for a pathologist to be involved is over here in integrating the data together and communicating it effectively to the clinical team. That we've tried to do with our report is to really integrate that data. So, we're integrating the molecular data with the morphology. We're integrating the variants back to the morphology, to the slides, to the molecular data, and then incorporating the clinical information into that and trying to communicate it back to the oncologist in a way that makes sense. Another thing we've done specifically with this test is we've interacted a lot with our local oncologists at the Swedish Cancer Institute in the development of the test and ongoing revision of the test. So some of the ways to make this work, first off, you need to optimize the assay just like we do with everything in the molecular lab. It's important to look at tumor enrichment, in other words, to get that DNA content up as high as we can. Some of the things we can do, for one thing, we can use fine needle aspiration. We find because the DNA samples are automatically enriched for tumor cells. You have less inflammatory cells and you have less stromal cells in an FNA sample. This has actually been proven. Usually you have someone on site to look at it and make sure it's adequate so you don't wind up with core biopsies that have no tumor in them. So if possible, you can use FNA. If not, you can, of course, microdissect slides, and you can do this with H&E sections or with cytology slides. One of the really interesting ways to do this is illustrated here. This is using this pinpoint solution. It's kind of like nail polish that you put on, and you just kind of draw the area and peel it right off. So you also can do paraffin cores. In other words, you take a little cylinder out of a paraffin block where you know the tumor is. Okay. Optimization of assays, tumor enrichment, mindfulness of DNA quality issues. Again, I had mentioned a minute ago that poor quality DNA will not work so well at a very low level. So some of the things we do to avoid that are looking at a gel, like a DNA smear of our genomic DNA and our amplified DNA. The picture that I'm showing you here, there's a big red circle showing you an area of DNA degradation. It's just a big smear. You don't see a sharp band. There also is a little area of RNA contamination down at the bottom. Both of these things can affect your assay. and they're important to look at. There are some ways that we can get around it. We can look at a different area of the tumor or a different block. We can put in more DNA if we think the quality is not as good. So we always assess this before we run our assay, and it's definitely something to keep in mind. The quality of DNA is really important. Ask about fixatives. Formalin does degrade the DNA somewhat, but we can still use it. And if you see here, the two that have formalin, the cyto-rich red and the formalin, are still fair quality for DNA. We still can use it. However, dissolution completely destroys the DNA. It's impossible to do PCR on decaled sections. And some of the other more unusual fixatives, such as B5 or buins, also will kill PCR. Pathologist participation in specimen procurement. So this means advising your radiologist or your surgeon about what would be the best sample, what would be the best way to handle it, and also, if possible, being there for the procedure in the case of an FNA. Also, pathologist involvement in the selection and processing of samples. So in other words, choosing the correct block, circling the correct area, and adequately assessing the area of tumor content. In mind, in specimen procurement, the pathologist provides guidance to the clinicians. The pathologist can provide an on-site evaluation and can actually triage the material. So you can assure that you have enough, you can assure that it's properly fixed, and you can preserve some of it for the testing that you want to do. So if you can do on-site evaluation, it's helpful. Unfixed material also can be very helpful. You have higher quality nucleic acids, less DNA is required, and there's less noise with NGS. Options for this include air-dried smears, alcohol-fixed smears. You can actually put this on FT8 cards, which are those little cards that they do for newborn screening, or you can, of course, freeze tissue. People are always fixated on the nanograms of DNA that you're putting into the assay and how many cells you would need to achieve that level. This chart shows you how much DNA you can expect per cell. The average pass in an FNA includes 50,000 to 100,000 cells per pass, so you could expect about 500 nanograms of DNA. Now, this is assuring that you have adequate extraction and that you capture all of those cells. But this is a good guide for how much you need to acquire. Is actually selection and processing of samples, looking at the actual slides. Again, when you're looking at slides, you need to differentiate which part of the tumor is invasive. You want to find the area that has the highest percent of tumor. Definitely no decalcification, and you actually want to assess for heterogeneity as well. So if two parts of the tumor look different, or if you know they have different expression on immunohistochemistry, like if part of your tumor expresses HER2 new and part of it does not, you probably want to test both areas. Of the percent tumor DNA is critical, and on-site pathology is a big advantage. Okay, our panel includes 60, we're going to stop for our second polling question here. There we go. This one, right? The following sample is not appropriate for NGS. Stop there. If decalcified bone marrow biopsy, you are correct. Decal kills PCR. You really to find tissue that has not been decalcified. So this is a way you can be helpful before receiving the sample and also afterwards. Decal is very bad for anything that involves PCR. All of these other samples are appropriate. The panel includes 68 genes, and this is a custom targeted MiSeq panel. We did design it in collaboration with our oncologists here at SCI. Compared to the standard MiSeq panel or the standard Ion Torrent panel, our panel has added coverage around the hotspot target areas, and we do have added gene targets. And I should also say that the genes that we know are really quite important and have known drugs that target them, we've expanded the coverage to include the entire exon region of that gene. So for KRAS, we include all exons. For EGFR, we include all exons. That we added, some of them are specifically related to leukemias and lymphomas because these tumors are not really adequately addressed in the common commercially available panels. We also added in genes that really just kind of came up between the time those panels were originally conceived and made available commercially. We also added in a couple of pharmacogenomic markers that are important to patients getting chemotherapy. Clinical scenarios we used in choosing our genes were built around the most common solid tumors and hematologic malignancies. I'll show in a second, and we do plan on updating this panel in a 6 to 12 month cycle. So in other words, we'll look at which genes we need to add. We'll look at whether or not there are any that don't really need to be there. And ultimately, I think we probably will progress to whole exome sequencing, I would say, within the next 3 to 5 years. And part of that just depends on increases in technology, when we can process the samples faster and when we can analyze more material. The tumor types that we considered in designing our assay and some of the genes that are commonly associated with those tumor types. We run the entire panel on every tumor, so it's not as if we're only genes listed here according to the tumor type, but this gives you some idea of the rationale we had for including some of these genes. So the first thing we do is look at the slide, identify the area for extraction, perform the DNA extraction, do the library creation, so this is adding the adapters and getting the sample ready to sequence, and then the massively parallel sequencing itself. This process takes about 5 days. We have the data analysis steps. This is when we actually have the data in the instrument. How do we kind of arrange it and get it out? There is a program within the instrument that helps us identify the variants. We go back through and we can filter using many different parameters and using a couple of other different software programs. And then we can create an edited VCF file that would contain all of the variants that we thought were real and that were present at an acceptable percentage. Our edited VCF file off to a collaborator that we have. We work with a company called Collaborex to obtain some clinical data regarding our variant in that tumor type. And see, a slide shows the Collaborex process. They basically aggregate data from many different sources, including both publicly privately held websites, also peer-reviewed literature, clinical guidelines, professional organization recommendations, drug trials, and cancer databases. They parse the data that they have, map it, and integrate it. The data is vetted by a medical advisory panel, including representatives from ASCO and CAP, and then they put that all together in a file that relates to our specific VCF file and our tumor type, and they send that back to us. Our system, designed here in-house at Symbiotics, can then take that information individually and put it in pieces that we can then go back and reevaluate. So a pathologist for every case will sit down and reevaluate every bit of information that comes to us from Collaborex, and we'll also look at the medical literature in general to make sure that nothing is excluded. Gather the most relevant parts of that information into a clinical report, and then the clinical report goes out to you. Report looks like. You can see up near the top we have just the diagnosis, the tissue, the original case number, and the tumor content. Then we have 3 categories. The blue box that says mutation summary is showing an actionable category, an applicable category, and unknown significance category. Every gene that has a variant that is shown to be significant will go in one of those 3 categories. Then down underneath, you see a summary of the gene, so what it does in the body, what it does in the tumor, and then the variant. So again, like what do we think this variant does to the protein, and what do we think the protein is doing to the tumor? see a paragraph in general about therapeutic relevance, followed by a list of drugs and a list of clinical trials. Each drug or clinical trial has a little box next to it. If it's a solidly filled in box, there is a drug for that tumor type targeting that variant that we are confident will work. If it is an empty box, there may be a drug targeting a related pathway that we have some reason to think may be helpful for this patient. If it's a half-full box, usually that means there is a drug targeting this mutation in a different tumor type. So in other words, it's kind of an off-label use. About our panel and a description of our legend there at the bottom. To be easily understood and directly useful clinically, we do provide an electronic version to our clinicians that has active links to the relevant literature and clinical trials. We think that our assay is affordable and reimbursable. Our list price is $2,400, which I think is pretty competitive for these types of things. We currently have a 14-day turnaround time. In each case is reviewed by a pathologist at multiple stages of analysis. So, in other words, from that very beginning step of looking at the tumor and looking at the slides and making sure that our results correlate back to the morphology, to the very final step of actually vetting all the clinical trials and drugs and making sure that the recommendations we're giving make sense, a pathologist is always involved with the case. Docs, which is a portal for our clinicians to get this very nice PDF report that we're sending out that has all of the active links. They also do many other things in this portal system, and this integrates our in-house software that we've designed to aid in analyzing these cases. Through your electronic medical record, you can get it through LabDocs. You can track the progress of your cases. Your staff can use the system, but each person would need to be set up in the system if you wanted to use it. In the remaining minutes here, I want to talk about clinical utility, because this is actually the most frequent and best question that comes up is, when should we be using this? I put this up here. This is not us, of course. This is the Cancer Treatment Centers of America, and I put this up there because it's ubiquitous. You hear it all the time on the radio, on the TV, everywhere. Have your tumor genome analyzed, and this is going to be a big advantage. So in part, I think it's true, but in part, I think we also need to be thoughtful and careful about how we're using this technology. Study actually published just in March in the Journal of Clinical Oncology from Dana-Farber, where the clinicians had access to genomic sequencing on their patients' tumors for free on every single patient. This was already done as part of their research program. The interesting part of this is that not all of the physicians wanted the information that was freely available to them about these tumors. Here it's kind of across the board. Many of them wanted it almost never, and some of them wanted it all the time. And most people were kind of hanging out somewhere in the middle at a mid-level. They thought they might want it on some of their patients, but not all of their patients. Interestingly, their level of comfort with this type of testing really related back to their knowledge about the testing and how comfortable they thought they would feel discussing the results with their patients. Probably the most telling statistic is the left of both of these graphs. The blue line is tier 1 alteration. So in other words, this is like your BRAF V600E mutation that there's a drug that works. This is going to have direct benefit to your patients. Only 85% of doctors thought that mutation should be discussed with patients, and only 70% of them thought that you should discuss a wild type BRAF result with your patient. So it's really kind of shocking, because this is information that's available that would help your patient. But this is a little bit of what we're up against in this type of testing. Oncologists, I think, reasonably so, are cautious and like to evidence-based care. And in this area, there's not always a clear trail of evidence leading to how to use some of this information. The variation in these results really related back to how much people knew about the testing and how comfortable they thought they would be explaining the results. So, one thing that we've tried to do is make the results easy to explain and to provide lots of interaction between us and the clinicians. And I think this is really critical moving forward, and it's definitely a role that pathologists need to feel comfortable in and need to be willing to step into. So the bottom line is this assay can provide treatment options for our patients, mostly in the form of clinical trials where they may not be apparent. So, just because you think that there may not be anything for your patient here, you don't really know until you do the test. Obviously, that's a gray area. It may not be reasonable at this time to test every single person, but on an individual level, I think it's entirely reasonable for our patients to want the test. That is that the technology is just outpacing the associated clinical research. So we can do things that, don't always have easy, straightforward answers right now. But that doesn't necessarily mean that we shouldn't be using it and that we shouldn't be trying to get more patients into those clinical trials where we can actually determine the information that's going to be helpful. Don't operate in a vacuum. If you're an oncologist, talk to your pathologist. If you're a pathologist, talk to your oncologist. We really need to work together to advance this field. And the fact of the matter is it's going to advance anyway. There are companies doing direct-to-consumer testing for germline mutations, such as this 23andMe, where people walk around with their genomes on their cell phones. And, patients want this type of testing, and they want it rightfully so. So we need to figure out how to make it happen in a clinically responsible fashion. And this is largely what we've tried to do here at Symbiotics in developing our test. If you do have questions about our test moving forward, this is me, again on the left, and this is my partner over here in molecular diagnostics, Dan Ben-Shu, who helped quite a bit in the design of our current assay and helps with the ongoing design and interpretation of this assay. So we both are available here for any questions that you might have about this testing or about our assay in particular. So at this time, I would like to open this up to any questions that people may have. I don't see any submitted, but if you have some, please go ahead and type them in and submit them through the system.
Okay, we're giving them just a couple minutes to do that. If there are any questions, I'll go ahead and just up on screen right now. If you're a PAML client and you like more information, you could please contact your PAML account representative. And for those who are not PAML, please contact Bev Meyer, and her email address is up there on the screen. So Dr. Berry also provided some additional resources, so those in your handout. And as a reminder, those of you who maybe now would like to download a copy of the handout, if you've closed that box that initially had the opportunity to download the handout, you can get that back by left-clicking up on File in the upper left-hand corner, and then left-click on Transfer, and you'll be able to download both the handout and also the evaluation. I want to remind you to, if you do want PACE credit, you do need to turn that in, but into your PACE certificate, even if you do not need PACE credit, please take time to fill out the evaluation. It's very helpful for us to get feedback. So have one question come in, Dr. Berry, that is, how does this test compare to the Foundation Medicine test?
That's a good question. The Foundation 1 test is a bit more far-reaching. It's also a targeted sequencing assay that looks at, I believe, somewhere in the 200 range of targets, and they also look at translocations and copy number variation. I would say it's much more far-reaching. It's more expensive, and it again is like a test in that they are just kind of looking at the slide and processing it through. I would say our assay is different in a couple of different regards. We look at a smaller set of genes and we cover them in a different way. I mentioned earlier what our rationale was for choosing our genes. Cheaper, and we do have the intimate involvement of a pathologist at every step along the way. So, so our pathologists are a lot more involved. But I think there are probably just different instances where you'd want to order the different types of tests. I think for patients that are quite advanced and maybe you're just completely out of options, you may want to go with the Foundation 1 test. But for most of your oncology patients, just looking for the reasonable therapies that are available to them in clinical trials, our test would be a good choice.
Thank you. We are in time, and there aren't any more questions. I would like to take this time and thank you, Dr. Berry, for taking your time and also sharing your expertise with us on the genomics and the next-gen sequencing. We really appreciate it. So,
Karen, I do see one more question.
okay.
Are the tests covered by insurers, including Medicare? So we've been doing this testing now for about 6 weeks, so we don't have a long experience with reimbursement, but we did do quite a bit of research into this beforehand, and other labs that have been doing this type of testing claim to 90% reimbursement rate, and that does include Medicare. So our stance as of now is that we are submitting this to insurance just like any other clinical test. We don't have any special requirements regarding advanced beneficiary notices or that type of thing. Again, we started doing this test, so we'll be able to assess our experience with it better in about 6 months to a year's time.
Thank you. Thanks, Dr. Berry, and I will leave up this slide and leave the webinar open for an additional 10 to 15 minutes if you'd like to download any of the handouts. So appreciate everybody's attendance and have a nice afternoon. Thank you for being